this video we will discuss about some of the strategies that are used to study the circular RNA. So this is the part of nucleus, and one of the strategies that people have used is to use this CRISPR-Cas9 system to cleave off some of the intronic sites that helps the RNA to make a circular shape, thus preventing the RNA to get into the circular shape, as the intronic sites that are flanking around that helps the mRNA to form a circular shape will be cleaved off by CRISPR-Cas9 system. Another approach that people used is to use this short hairpin RNA that with the addition of this CRI will form a short interference RNA that is shown here, and this short hairpin will form a short interference RNA, and thus this short interference RNA will have the ability to cleave off this circular RNA so that they can understand the effect of absence of the certain circular RNA on the cells or on the experiment, whatever the kind of experiment are designed. The third system that is shown here is of this CRISPR-Cas13 system. So what it will does is it will cleave off this circular RNA that are in the cytosol, and it can directly cleave off the circular RNA after it is formed. Whereas instead of this, when we are using this CRISPR-Cas9 system, here the intronic part of that flanking around this intronic part is cleaved off, thus there is no formation of circular RNA happens. Whereas in case of CRISPR-Cas13, it will cleave the circular RNA after it is formed. And another way to another way to study circular RNA is by overexpressing circular RNA through the plasmid expression. So the plasmid of interest will be transfected inside the cells. Then it is overexpressed by using the machinery of the host. So these are the different strategies, or we can say some of the strategies to study the effect of circular RNA on the host. And these strategies can work on like this CRISPR-Cas9 system will work mainly on the circular RNA that contains the intronic part. However, for circular RNA that only contain the exons, this CRISPR-Cas13 system will work. And whereas for using this short hairpin RNA with the help of CRI, it will form a short interference RNA. It will cleave off the circular RNA that are of interest, that is designed or that is been targeted in this short hairpin RNA. So this is, and this for this another kind of circular RNA, that is exon intron circular RNA. This can be studied using both of these strategy, that is CRISPR-Cas9 system and CRISPR-Cas13 system, as it contains both the intron and the exon part. So whereas this circular RNA can be studied or manipulated only by this CRISPR-Cas9 system, or mostly by CRISPR-Cas9 system. However, some modification in this CRISPR-Cas13 system might also be able to work on this circular RNA that contains only the intron part. So these are some of the few strategies that are mentioned here. If you need to understand all this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.